Hello, dear students. I welcome all of you in this session, your semester fifth. In this lecture, we will discuss about the basic introductory part of your syllabus, that is your semester fifth, microbiology, and paper name is microbial genetics. So here we have to discuss about the genome of prokaryotic and eukaryotic organisms, different methods of transformations, mutations. These are nothing but the content of your syllabus. So in this, we will basically discuss some of the basic definitions with respect to the mutation and introductory part of mutation. Here, a mutation is a change that occur in our DNA sequence, either due to the mistakes when the DNA is copied or as the result of the environmental factor. So nothing but it is nothing but the changes in the DNA sequence, and due to that mutation occur. Mutations in egg or sperm cell are called as germinal mutations. This type of mutation may result in an individual offspring of all of whole cell carry the mutations, or whose cells carry the mutations, which often confirm some serious malfunctions, as in the case of human genetic diseases such as cystic uh, fibrosis. So these are nothing but the when mutation occur in the germinal cells, it has carried out throughout that cell and leads to the uh, serious malfunctions of the body. Also leads to the different genetic diseases like cystic fibrosis. Mutations result either from the accident during the normal chemical transactions of DNA during replication, or from exposure to high energy electromagnetic radiations, for example, ultraviolet light or X-rays. So these are nothing but the different high intensity light or high energy light. When the cell is exposed to that light, leads to the changes in the DNA sequence of a particular cell and leads to the mutation. Mutations are random changes. They are expected to be mostly deleterious, but some may be beneficial in certain environment. So, with respect to that DNA mutation, we can also see some repair mechanisms are also carried out in the cell. So, we'll discuss one by one. Basically, in the broad sense, the term mutation refers to all the heritable changes in the genome, excluding those resulting from incorporation of genetic materials from other organisms. A mutation is an abstract qualitative or quantitative change in the genetic material of an organism. So these are nothing but the basic definition of mutation. Here we can see mutation have different types like intragenic or intergenic type of mutations. So basically, so firstly, the first type of mutation is nothing but the intragenic mutation, or we can also call it as a point mutation. It involves the changes, or we can say alteration, in the normal base sequence of a DNA molecule within a gene. These changes modify structural characteristics or enzymatic capability of an organism. So it is nothing but the definition of intragenic mutation, and we can also call it as a point mutation. It is carried out a particular point in that particular DNA sequence, or you can say gene. Another type is nothing but the intergenic mutation. It involves changes in long region of DNA, that is many genes. This includes addition or deletion of segment of the chromosome. So here long sequence of the DNA are either removed or added into that particular DNA sequence and leads to the intergenic mutation. So these are nothing but the basic idea about the mutation. Next part we'll discuss in next lecture.